I read an interesting report from NASA. Originally, the space station used just to dump the human waste overboard, which means we had all this stuff circulating in space, adding millions of organisms into outer space. But now they encapsulate the solid waste into stainless steel containers and launch them back into the Earth's atmosphere. So be careful the next shooting star you really wish for. You may get more than you bargained for with it. Anyway, these unusual organisms, it turns out brewer's yeast can do this itself. Not in terms of the radiation, but it can just naturally reassemble pieces of DNA that we insert in it. And we started by inserting the 4 quarter molecules in there, and it just basically instantly reassemble these quarter molecules into the entire genome. We have a paper coming out this week in PNAS doing this now with 25 much smaller molecules, and we just put the 25 smaller pieces into yeast, and it reassembles it. You have to design them properly so they overlap. Just instantly reassembles these into the entire microbial genome. So earlier this year, we reported on, this is the largest human-made molecule of a defined structure. It's over 3 million molecular weight. It's over 582,000 base pairs. And if you're printing out its sequence at a 10 font with no spaces, it would take 147 pages just to print the sequence. We resequenced it, and it was 100% accurate for what we designed. So the big question we're struggling with now is how do you boot up a synthetic genome? Oh, by the way, here's a picture of it. It's such a large circular molecule, we don't even need an electron microscope to see it. So how do we boot up a synthetic chromosome? And I think this is a study we published last year, is one of the most important studies thus far in my career, where we actually changed one species into another by replacing its software, its genetic code. And I think this has profound implications for evolution that we'll get to. Let me explain what we did. We isolated the genome from M. mycoides. We added harsh enzymes to it to destroy any proteins associated with it. We added a couple extra genes to it, one set that would turn the cells bright blue if it was expressed. We then inserted that DNA into a donor cell, M. capricolum, which is a related organism. But it turns out we knew a little bit more through the power of genomics. We knew that the donor chromosome contained multiple restriction enzymes. Now, my friend and colleague Ham Smith received the Nobel Prize in 1978 for discovering restriction endonucleases. I always appreciated them as a tool, but I never truly appreciated their evolutionary significance till we did this experiment. We also had the sequence of the capricolum genome, and we knew it had a number of restriction enzymes. But we also knew 2 other things. We knew those enzymes in M. capricolum did not recognize anything in the sequence of the mycoides genome. In contrast, the ones in the mycoides genome did recognize the capricolum sequence. So through this sophisticated artwork, I'll show you what happened. We inserted the chromosome into the cell, and as with the viral DNA, the cell machinery recognized this, started making all the proteins, including the restriction enzymes. The now newly synthesized restriction enzymes recognized the parent chromosome as foreign DNA and immediately chewed it up. We're now left with cells. The only difference is they had a new operating system that we had inserted. In a short while, we had bright blue cells. Even more stunning, when we looked at every protein in the cell, every protein that was there before was gone, and all the proteins that define the species now were coded for by the new software that we put in the cell. So we completely changed one species into another. Now, was this some strange thing of just the work that, wizards do in the laboratory? It turns out we look back at genomes that we'd sequenced, and this happens all the time in evolution. So everybody thinks of evolution, or most people do that even think about it, in terms of point mutations and then selection. Early on, Rita was a proponent for having the cholera genome sequenced. Carl Woese actually argued strongly against it because its 16S RNA was essentially the same as E. coli. So he said there's no point sequencing the genome, we know what it looks like. Well, my late mentor, Nate Kaplan, the key thing he taught me was never talk yourself out of doing an experiment. So we sequenced the cholera genome, and guess what? It had 2 chromosomes, not just one. One was like E. coli, and the other one looked nothing like it at all. And we see this over and over again. The organism Deinococcus radiodurans I told you about has 4 chromosomal elements. All you have to do is have a mismatch in the restriction enzymes, and if cells fuse or they take up DNA, you can add thousands of new traits instantly in one step in evolution. We don't know how many times this has occurred. We don't know how many times the experiment, like I showed you here, occurred where after taking up a chromosome, the parent chromosome was destroyed. I think this is going to be a major factor in understanding evolution. So why do this? Aside from trying to understand basic cell structure and evolution, we've been thinking about these processes for some time, and we thought they might have a role in maybe solving some of the problems. You heard from our 3 distinguished winners about population growth. And I use the same statistic. I was born in 1946. There's now 3 people here for everybody that existed the year that I was born, and soon there'll be 4. We don't have enough food, clean water, a shelter for those that exist now, as you so nicely heard from the prize winners. Also, as we're referred to with increased economic growth, yes, you have a lower population expansion, but you have an exponential increase in the amount of energy demanded by those populations. Food availability is an increasing problem, particularly as we use so wisely cropland for producing ethanol in this country versus food. And everybody is pretty much aware in this group of the potential effects of climate change on future droughts. So where do we go from here? I'd like you to introduce a new concept to you. I view these 20 million genes we have in our database as the design components of the future, much in the same way that resistors and capacitors and transistors were in the 40s and 50s. Only instead of having a handful of components, probably within 5 years, we'll have 100 million, maybe more in the data set to work from. At Synthetic Genomics, we actually have software for designing new species. So just in case you didn't have enough biodiversity, we can make more. It's probably not what you had in mind, but we can actually design these organisms for very specific purposes, including to make sure, as with every molecular biology experiment over the last several decades, not to grow outside of the lab or survive outside of the intended conditions. We're working on designer fuels. The first generation fuel is obviously sugar to ethanol. I think ethanol is great that it mixes with water, particularly if there's ice associated with it. But it's a lousy fuel. It's not very high energy, and the fact that it mixes with water is a problem. But using microbial pathways, we can make any chemical starting with sugar. And there's multiple ones coming out from various groups. We have some new fuels that have low freezing points, minus 116 degrees centigrade, much higher energy content. But the fuel industry doesn't really want more alcohols. We need something that's truly different. And also, we don't need things that compete with food for fuel. So we've been working on using CO2 as a feedstock. We have plenty of energy in the form of sunlight. We only need a tiny fraction of it for all, even transportation purposes. I've argued this a few million hectares would be a good use for Nevada, perhaps Arizona. When we look at various plants, they're not very productive. The best being palm oil with about 600 to 700 gallons per acre.